Hello. Okay. Now we're moving on to finishing off chapter 2. This class, hopefully, this class session will go through the end of chapter 2 and we'll start chapter 3. Okay. Chapter 4. We'll skip chapter 3 for now. Okay. Okay. One second. I don't know where my file is. Chapter 1. Okay. Let's pick it up. So share screen to slideshow. Oops. Okay, so we ended with the nucleus, right? The most important organelle, obviously. So now we're going to talking about DNA, okay? I'm sure all of you guys know DNA and you can probably picture the double helix structure of DNA, okay? Something that looks like this, okay? So you have 4 nucleotides, A's, T's, G's, and C's, and A's always attached to T's and G's always attached to C's, okay? That is the idealized structure of DNA. That's not really how DNA exists inside the cell. Okay, DNA is usually packed with protein. There's a bunch of protein that surround it. Okay, the proteins are called histones. The DNA plus the proteins together form something called chromatin. Okay, each cluster of DNA and histone proteins is called a nucleosome, and you'll see a nucleosome looks like a little ball. So little balls, okay, and then these little, so they look like beads on a string. That's the term that's always used to describe them, okay. And these little strings get strung together in a loop, okay. This is kind of like the way you loop telephone cords and stuff between the receiver and the base of the phone before we all have cell phones and stuff. That type of coil is what you see here, okay? Okay, and that's chromatin, okay? The chromosomes, okay? I'm sure when you picture the term chromosome, you think of something that looks like X, okay? And it does look that way sometimes, but not usually. So this is the X. Okay, this is a chromosome. The chromosomes only form when the DNA is actively dividing, okay, or when the cell is actively dividing, okay. Usually the chromosomes just spread out through the cell, and you can't really see, like there's no structure, okay. Okay. So let's go through the cell cycle. All right, and okay. So like I said, excuse my really crappy drawing. So draw a big circle. Okay, so here is my really big circle. Okay, so this is a cell cycle. Okay, one cell goes in, 2 cells come out. Okay, so now we'll draw something else. Okay, the first thing when we discovered microscope, we can look at cells, but what we didn't see was what was actually going on. In fact, there was only one time where we could see what was going on. And this was the mitotic phase, or M phase. Okay, this is where one cell divides into two cells. Okay, so the mitotic phase is where, or M phase, is where one cell divides into two cells. Okay, and we can see that because we can really see one cell turn into two cells. And we can see the nucleus disappear and all this other stuff that goes on in this phase. But we couldn't see anything else. Okay, so what happened was, okay, we're going to call okay. The rest of the cycle interface. Okay, the rest of the cycle is called interphase because it's literally between mitotic phases, interphase. Okay, and then our technology got better, our science got better. We could see a little bit more and understand a little bit more about what happened to the cell. And so the next thing we found was another phase of the cell. Okay, this phase, we'll label it in orange here, is called the S phase. Okay. The S stands for synthesis. Okay. Okay. The S phase is where DNA replication actually occurs. Okay, so before cell division occurs, the DNA has to replicate itself. The DNA replication forces a lot of things to be synthesized. You have to make a bunch of stuff before or during DNA replication and before DNA replication. So S phase, okay, synthesis phase. Okay, so now you can see two phases and you can see two gaps. Okay, so gap 1 or G1. Okay, this gap became growth phase 1 or G1. Okay, it's initially called gap phase 1 and then got retrocon to growth phase 1 because what we realized was lots of cell growth. Okay, a lot of growth occurs right here because like literally right here they just divided into two cells here, and so one cell is about half the size of the original cell, so it has to grow back to the original. And if this is gap phase 1, then obviously this is gap phase 2 or growth. Okay, growth phase 2 is the prep for mitosis. Okay, mitosis requires a lot of proteins and a lot of enzymes and a lot of energy to be made. And during the phase, okay, you don't have time and energy to make anything. So you literally need to make everything in growth phase 2, and then go through mitosis all in one big, command. So all the prep for mitosis occurs here. Okay. All right. So that is the 4 stages of the cell cycle. Okay. Okay. So that hopefully I explained all these 4 phases. Now let's look in more detail about mitosis and cytokinesis. Okay, so first thing I want to point out, the M phase is the mitotic phase, not mitosis. A lot of people confuse mitotic phase with mitosis. Mitosis is one part of the mitotic phase. Okay, mitotic phase splits into mitosis, which is the division of the nucleus. Okay, and cytokinesis, which is a division of the cytoplasm. Okay. All right. The first stage of mitosis, okay. Okay, is prophase. Prophase is the first and longest stage of mitosis. Okay, so let's look at prophase. So what I'd like to show is how things change. Okay, so if you look from here to there, okay, so this is the pictures and this is the model, right? So here, okay, you have interphase, here you have early prophase. What you realized was here, okay, between here and here, okay, so interphase, the DNA is spread all over the nucleus. So this is all uniform light blue. The light blue is the DNA, okay? The dark blue, when you see it, is actually the nucleus itself. Okay, so you can see the light blue uniformly spread here, and so you can see the DNA is all over the place. In early prophase, the very first thing that happens is the chromosomes condense. Okay, so imagine you were, living in your house. Your stuff is spread all over the house relatively equally. Okay, and then you're going to move into a new house. The first thing you do before you move on to the new house is pack up all your stuff. Put all your stuff into boxes so they don't get lost and they're easier to transport and they don't break, etc., etc. Okay, the nucleus does the same thing. It takes all the DNA and packs them neatly into chromosomes. Because then you can move chromosomes relatively easily. Okay, so now let's look at so that's why when you look at this picture, you can see the DNA is more condensed. You see areas where there's no DNA. Okay, and you can see that the DNA looks like little strings. Okay. If you go from early prophase to late prophase, if you look at the DNA, okay, the DNA is now just spread in the cytoplasm. The nucleus has broken up, okay, and that's the second key feature that happens in prophase. Is that the nucleus disintegrates. Okay, you don't have to distinguish between early prophase and late prophase. Okay. All right. The second stage of mitosis is metaphase. Metaphase is the easiest stage to actually understand. Okay. All the chromosomes line up in one nice little row. That row is called the equilateral plate or the metaphase plate. okay? And then you have little spindles, okay, that attach to the middle of each chromosome, okay? That place of attachment is called a centromere, okay? The spindles are actually caused by the centrosomes. Remember yesterday, I said the centrosomes were the place where all the microtubules attached. And you can see that here. This is the place where all the microtubules attach. But now the microtubules have a different job. They're coming to the center of each chromosome, okay? And you can see there's two of them now, and they're attached to both sides. So now I'm in metaphase. The next step is anaphase, okay? Anaphase, I just pull the cells apart, and you can see the little V-shaped thing here, okay? Okay. And then last, okay, is telophase. Telophase is almost the opposite of prophase, okay? Telophase, you have a new nucleus forming. Remember, the nucleus is disintegrated in prophase. So in telophase, you have a new nucleus forming, okay? Here, you also have the DNA unraveling a little bit. After it's been packed, it needs to now unravel so it can do more stuff. Well, you need to unpack before you can actually do anything inside your new house, right? So the cell has to unpack its DNA before it can do anything. Okay, so that happens at the end of telophase. Okay, cytokinesis. Cytokinesis is pretty simple. It's much simpler than mitosis. Cytokinesis, I just make a ring around the edge of the cell, and I bring in the ring closer and closer and closer, like a drawstring bag. When you pull the string, the hole gets smaller and smaller until it just pinches off and you separate. Okay, that makes two new cells. Okay, it's not a very careful procedure. You don't really care if there's like 10 mitochondria on this side and 20 mitochondria on that side. As long as you have some mitochondria on either side, you're fine. If you have some ER on either side, you're fine. If you have some ribosomes on either side, you're fine. So it's a very easy procedure. You just got to pinch it in half and worry about everything else later. Okay. Okay. So here. Okay. So prophase and prometaphase are both considered prophase in this next book. So don't worry about that. Okay. Okay. You don't need to know about plant cells, right? So don't worry about that. Okay. So the developmental aspects of cells. Okay. So cells are different shaped, obviously, and they have different arrangements of organelles. And the way they're shaped and the way their organelles are arranged are ideal for that particular cell type. Okay, so we can look at some of them. Okay. Okay. So let's take a look. These are fibroblasts. You can see they're long cells, so they make long fibers. Okay. These are erythrocytes. Okay. Erythrocytes are also known as red blood cells, and you can see they don't have. They're the only cells without a nucleus, so they have no nucleus, so they don't need. So there's more room for hemoglobin. These cells are flat disc shaped, so they fit through the blood vessels very easily. The circular formation allows them to fit through very easily. The thin thing in the middle, the divot on both sides, okay, the biconcave shape allows more surface area for gas exchange. Okay, epithelial cells, okay, are going to form these little squares. They're going to form linings. Okay, they will separate one thing from another thing. So what's going to happen is one, they have to line up all in one nice little row, and two, there have to be no gaps between them. So you can't use circles. The circles leave gaps. So you use squares or hexagons. Those don't leave any gaps between the cells. Okay, so cell shape is determined by cell function and vice versa. Okay, and you see muscle cells are long, fat cells are just, this is a fat cell, so literally this is the nucleus and this is the fat. Okay, and that's it. There's nothing else to that cell. Okay, so we'll skip the rest of this. Okay, we'll skip the aging process thing. Okay, so I'm gonna stop this for part 1 right here. Part 2, we're actually going to chapter 4. Okay, we're skipping chapter 3. So